Hello everyone and welcome, and hopefully also welcome back now to our second aberrations Instruments Summer Symposium. My name is Janina Hanne and I'm the Scientific Relations Manager at aberrations Instruments, and I will guide you through today's program. Our topic for today is Adaptive Optics STED Microscopy, and we have in total 4 excellent speakers today. First, Mary Grace Velasco and Alex Egner will give a short talk each about their research on adaptive optics in STED microscopy. Next, my colleagues Matthias and Gero will introduce us to how we have implemented adaptive optics into our own confocal and STED microscopes at aberrations Instruments. And then they'll show us in a live demo how adaptive optics improves STED and confocal imaging quality in thick samples. Please use the questions tab of this webinar software to ask any questions already during the presentation, so that we can start right away with answering them in the Q&A session. And also, please don't forget to register for our next symposia. STED Paint is next week, and life-size STED microscopy in 2 weeks. Now I'm really happy to introduce to you our first speaker, Mary Grace. She's a physicist by training and worked with Jörg Beversdorf at Yale for her PhD, in which she pushed the boundaries on how deep you can image with STED microscopy in living mouse brains. I'm super happy that end of last year, Mary Grace joined our aberrations Instruments America team as an application scientist. The title of her talk is Wavefront-sensing and aberration correction for in vivo STED microscopy. Mary Grace, I'm looking forward to your talk. The stage is yours.
Hi everyone, it's me, Mary Grace. I hope you're all doing well and logging in from somewhere safe. And of course, thank you for taking the time to be with us today, despite everything that's going on. Now, as Janina mentioned, I'm an application scientist for Abberior Instruments America, but today I'm going to be reprising my role as a graduate student and presenting my PhD work on wavefront sensing and aberration correction for 3D STED microscopy. So that said, I'd like to mention that all the work I will be presenting here was carried out in the Buersdorf lab at Yale University under the mentorship of my PhD advisor, Jörg Buersdorf. Okay, so let's get started. Optical aberrations, what exactly are they? Well, you can think of optical aberrations as distortions or deviations of the shape of light from what they ideally should be. So in an ideal microscope, you want your light to be perfectly straight or flat, or if it's being focused, as it is here after the lens, you want it to be spherical. However, due to imperfections in your optics, or more importantly, because of mismatches between the refractive index of your objective immersion medium and the refractive index of your sample, the shape of your light gets distorted or aberrated. And it is these aberrations that compromise your microscope's ability to focus light correctly. Now, remember that in a STED microscope, you now not only have to worry about the excitation beam, but about the depletion beam as well. And if the depletion beam becomes aberrated, the donut shape, which is crucial for effective super resolution, will get all wonky and it won't be able to squeeze down your fluorescence in the way that you want it to. Thankfully, for 2D STED experiments, the 2D depletion donut that is generated by this vortex phase pattern here is relatively robust to aberrations, meaning that it can maintain its donut shape pretty well, even in aberrating samples. So you can do 2D STED tissue imaging experiments at about conventional confocal imaging depths with relatively little issue. Unfortunately, the same cannot be said for the 3D STED donut that is generated using this top hat phase pattern here, which is actually extra sensitive to aberrations. And basically, the way that this focus shape responds to aberrations is that you get depletion light leaking into what is supposed to be a perfect zero intensity in the center. And the effect of that, of course, is that you now have a depletion focus that switches fluorescence off everywhere, rather than just confining it to a subdiffraction limited size. And this effect applies even when you're combining this Z depletion donut with the XY depletion donut for optimal depletion in all three dimensions. So because of that, optical aberrations can pose a major challenge when doing deep tissue STED imaging, or even just deep tissue imaging in general. Now, when discussing aberrations, you'll often hear the term wavefront, which you can think of as the shape of light if you were to take a cross-section right through it. And to help visualize and characterize the aberrations present in a wavefront, it is a common practice to decompose it into a series of Zernike modes, the first 14 of which are shown here on this slide. Now, you can think of the Zernike modes as different aberration types, and they can be combined in different amounts to generate any arbitrary wavefront shape. Ideally, the shape of the beam should be flat, such that the amount of each of these aberration types is 0. But when imaging in tissue, this, of course, is rarely the case. Okay, so now the first trick that we can play to minimize these aberrations is to use an objective that is equipped with a correction collar. So we all know how these work. You simply turn the collar to adjust the lenses inside the objective, and in this way, you're able to optimize your image quality. A second trick is to use an objective lens with the appropriate immersion medium. Now, as I mentioned previously, aberrations come from refractive index mismatches between your objective immersion medium and your sample. Now, the average refractive index of some biological tissue is close to that of water. And I should specify here, of course, untreated biological tissue. So for these applications, a water immersion objective lens, despite its lower NA, can actually be a better match. Higher NA objective lenses, on the other hand, these rely on oil, which works fine and is actually better for imaging cells, thin tissue sections, or samples mounted in an index-matched medium. But for deep tissue imaging in untreated specimens, the higher refractive index of the oil can actually start to have a detrimental effect. Of course, these aren't the only two options. As an intermediate between water and oil, silicon oil can also be used, for example. The important point is that your choice here will depend largely on the refractive index of the sample that you are trying to match. Now, by simply choosing the right objective lens, you can already minimize the bulk of the spherical aberration that is induced. And this is a great first step. However, there are other aberration modes to consider. Also, more importantly, there are local variations in refractive index that can induce additional aberrations that vary from sample to sample and even from field of view to field of view within the same sample. And so to address these, we need a more active method of correction. And this is where adaptive optics comes in. In adaptive optics, you use a special optical element like a deformable mirror, for example, which is able to modulate the phase of light on the fly. And now phase modulation is now just a fancy term for adding aberrations in a controlled way. The basic principle is that you manipulate the shape of the reflective membrane on your deformable mirror by sending signals to the individual actuators here. And if you can get the correct membrane shape, you can, in effect, pre-aberrate your light in such a way that the aberrations from the deformable mirror and those from the sample cancel each other out. And what you are left with then are your aberration-corrected excitation and depletion foci at the sample plane. And with those, you can then proceed with your aberration-free imaging. But the question is, what is the correct membrane shape? And how do we know how much correction we actually need? Well, one method, which I won't go into much detail, is called indirect or metric-based aberration correction. And for this method, you need a reliable image quality metric. And based on the feedback from this metric, you converge on the optimal shape of your deformable mirror membrane over multiple imaging iterations. Now, another method you can use, which is the one I used for my project, is called sensor-based aberration correction. And here, you use what's called wavefront-sensing to directly measure the aberrations that are coming from your sample. This method relies on having a guide star or a point source of light in your sample. And as described in these two references here, in the 2-photon microscopes that are typically used for deep tissue imaging, a guide star is provided essentially for free by the same volume of excited fluorescence that is typically used for imaging. Now, the fluorescence emitted by this guide star on its way back through the system acquires the same aberrations as those of the incoming excitation light. So when the fluorescence is coupled out by a dichroic mirror, it can be sent to a wavefront sensor like a Shack-Hartmann, which then measures the different aberration modes that are present in the aberrated beam. Then, once you have these aberration values, you can send the negative of these values to the deformable mirror so that a perfect beam and a perfect focus is recovered at the sample plane for aberration-corrected imaging. Okay, so for my project, I actually built a 3D STED microscope that uses all the elements that I have described thus far. It has a water immersion objective with a correction collar, which I have to admit I set for the correct cover slip thickness and then proceeded to forget about. The system also has an SLM to shape the depletion beam for STED. It has a deformable mirror for aberration correction, a Shack-Hartmann for wavefront measurements, and finally, it relies on 2-photon excitation both for imaging and also for generating the guide star that is used by the Shack-Hartmann. Now notice here that the wavefront sensing and imaging actually occur in two different channels. The wavefront sensing is in the green channel, and the imaging is here in orange. And the two channels are spectrally separated using a dichroic mirror. And so the imaging routine proceeds as such. First, you start by acquiring a Shack-Hartmann spot diagram, which is what's used to then reconstruct your aberrated wavefront and to decompose it into its constituent Zernike modes. Using these Zernike modes, you then calculate the appropriate correction to apply to your deformable mirror. And from a user perspective, it should be mentioned that these two steps, this analysis over here, you won't see it. It's all happening in the background. Then next, optionally, you can first judge whether the image quality from this correction is good enough for you. Or you can proceed to skip the step as I did for most of my experiments and then just proceeded to acquire an aberration corrected image. Okay, so now comes the fun part. So our first proof of concept application to show how the microscope works was the STED imaging of fixed mouse skin tissue. And here we were looking at the 3D organization and structure of chromatin in keratinocytes or in skin cells. So the tissue we used was from a histone 2B GFP mouse that was then stained additionally with an anti-GFP nanobody conjugated with the dye ADO594. We used the GFP fluorescence for our wavefront sensing, so it's not actually represented in the image here. What we're looking at is actually the ADO594 signal. Also, as this was a test for our aberration correction routine, we deliberately mounted our tissue in a medium that was not matched to our water immersion lens. So the STED stack shown here was acquired about 62 microns below the surface of the skin tissue, and as you can see, it's not exactly a very good image. However, if we do our aberration correction, we can recover our signal to noise and now actually clearly resolve the chromatin structure. And if we take a YZ slice here, we can do a similar comparison, this time in emphasizing the improvement using a YZ view. I should mention here that for STED, we only used the top hat pattern for depletion along the z-axis, since we didn't really need too much lateral resolution improvement to resolve the chromatin structure. And if we include here the corresponding to photon image, you can see more clearly that axial resolution improvement that we were going for. Comparing these two images here, you see that without the STED effect, the structures are stretched out in Z due to that cigar shape of the diffraction limited point spread function. If you're simply interested in what caused the bad image quality in the first place, you can actually get that information from the Shack-Hartman. The Shack-Hartman gives you a breakdown of the aberration modes that were present in this particular field of view of the tissue. And as you can see, since we were deliberately mismatching our immersion medium and our sample mounting medium, spherical aberration is the dominant aberration mode being corrected. Nevertheless, there are still other modes that were also corrected, which without adaptive optics would be largely ignored by your correction collar, for example. Our second application was the imaging of astrocytes in fixed mouse brain tissue. And so here the tissue that we used was harvested from a wild type mouse, and it was stained with an anti-GFAP antibody conjugated with, again, the dye ADO594. The tissue section was then mounted in PBS containing fluorescein. And again, we used the green fluorescence for our wavefront-sensing, so it's not actually represented here. What we're looking at is the ADO594 signal. And again, for STED, we only use the top hat phase mask for fluorescence depletion along the axial direction. Now, the center of the STED image stack was located about 164 microns below the surface of the tissue section. And without aberration correction, we see that there's a drop in the signal to noise of the image that prevents us from seeing the thinner astrocyte branches. With the correction, however, these details are largely recovered. And again, for completeness, I show here the corrected 2-photon image where you see the blurring of the thinner astrocyte branches, especially in the axial direction. Okay, so now our final proof of concept application was the imaging of dendritic spine dynamics in a living mouse. And for these experiments, we actually wanted to label the live neurons with the STED-compatible red organic dye ADO590, which, because it is an organic dye, it's brighter and more photostable than the red fluorescent proteins, which would otherwise be used for this application. More importantly, this dye is cell permeable and live cell compatible, so it is ideally suited for in vivo STED applications. To label our neurons with ADO590, we started with a mouse that expressed both cytosolic GFP and the protein tag halo in a subset of its neurons. And to get this, we used AAV virus injections. For imaging, we first attached a head post to the mouse, performed a craniotomy, and applied the ADO590 dye solution to the brain surface. And because ADO590 is cell permeable, the molecules can actually enter the individual neurons. And because the molecules have a chloroalkane, which makes them reactive to the expressed halo tags, this allows them to bind specifically. After the labeling, the craniotomy is sealed with a glass window, and the mouse is then ready for imaging. So here I'm showing an aberration-corrected 3D STED image stack of a neuron in a living but anesthetized mouse, which went through the labeling procedure I described on the previous slide. For STED, we used both the 2D and the 3D STED donuts combined so that we got optimal depletion in all 3 dimensions. And the center of the stack was measured to be about 76 microns below the cortical surface. Now, as the stack rotates about the x-axis, you clearly see the 3D structure of the neuron and its spines, and none of these details are blurred in the axial direction as they would be if you were to use a standard 2-photon microscope. Now, let's zoom into the spine over here. And you can see here that it actually bends or goes in and out of any given imaging plane multiple times. And these details or these changes in orientation are beautifully captured by 3D STED. Now, of course, the key advantage of this technique is the potential to do long-term imaging of the same structure over a certain period of time. And so to demonstrate this, we actually imaged the same area from the previous slide 2 days later. And again, we can zoom in on this, this spine over here. And we can compare it to its corresponding two-photon image. And as expected, without the super resolution capabilities that are provided by the STED effect, you see that a lot of the details are lost and blurred out, especially in the axial direction. And last but not least, focusing on just the day 3 images over here, we can zoom back out to the overview images and emphasize the 3D effect just one last time, just by rotating the stack about 90 degrees. And that, ladies and gentlemen, is my work on aberration-corrected 3D STED. I think I am close to my time. So very quickly, a big thank you to the Viewersdorf Lab family back at Yale, and of course to our collaborators. Thank you all for putting up with me for 5 or so years. I guess I will take any questions at the Q&A session following the next talk. But if we don't get to your question, of course, feel free to reach out to me here directly. Thank you very much, and I'll see you guys at the Q&A session.
Great, thanks, Mary Grace. That was an excellent talk, and you really showed amazing STED images. Actually, we got quite a few questions. So the first question is, can you comment on any phototoxicity or photo damage during the in vivo experiments?
Yeah, of course. We actually have not yet performed particularly quantitative tests for photo damage and phototoxicity for these experiments, though of course I definitely agree that such experiments should be included in any follow-up studies. But I will say this, there were a few design features that were incorporated into the microscope that correspond with published guidelines for minimizing photo damage specifically for live cell STED experiments. And for this, I'm referring to a paper also from Jurg's lab. It's a 2018 Nature Methods paper where a colleague did carry out quantitative photo damage experiments for this. And in their recommendations, they include the use of fast laser scanning and also the use of far red excitation and depletion wavelengths, both of which were key features of the design of our microscope. Sorry that it's not a very quantitative answer, but we also found it promising that during our in vivo experiments, especially on our multi-day ones, whenever we image the same area again after a couple of days, we found it promising that the same spine could be found. But yes, I definitely agree that included in the next steps should be some sort of quantitative photo damage experiment.
Right, thanks for this answer. Next question. Very nice work. I wonder how fast will be for a single correction and how large field of view is effective for the single correction.
Yeah, so in terms of the speed, a single correction takes the same time as a single image acquisition. So essentially, it's one image acquisition for the correction. You correct, and then you take your final image or your final image stack. And I should say that for the stacks that I took, I used a single correction that was at the middle of the stack. Regarding how large the field of view a single correction would be effective for, that is actually an excellent question. That will vary from tissue to tissue and was something that you kind of like have to test out for yourself given your application. And that's simply because the refractive index variations for any given tissue will vary. And so that you kind of like have to test beforehand before you do any imaging.
Great, thanks. Final question for you for today. Can fluorescent proteins also be used for the in vivo STED experiments?
Yes, of course. However, for most STED experiments, we always advise to use organic dyes over fluorescent proteins whenever possible, simply because they're just brighter and more photostable. So in general, we always say that STED microscopy uses the same has the same dye requirements as confocal, and as with confocal, you always want to use bright and photostable dyes. The difference is that with STED microscopy, this brightness and photostability requirement is just a little stricter, and so you'll always want to use like very bright and very photostable dyes for STED whenever you can.
All right, thanks a lot, Mary Grace, for joining us today.
You.
It was a pleasure having you here. And now let's move on to our next speaker. As the next speaker, I'm happy to introduce to you Professor Alexander Egner. Alex is the director of the Laser Lab in Göttingen and heads the Department of Optical Nanoscopy there. In his early career, he worked with Stefan Hell and contributed to the early development of STED microscopy. And additionally to STED microscopy, Alex worked on 4-pi microscopy, palm storm microscopy, and related concepts using photo switching of fluorophores to achieve optical resolution on the nanoscale. Now, he and his group are not only focusing on basic research in the field of super resolution fluorescence microscopy, but also on the practical implementations. Alex, thanks a lot for joining us today. I'm really looking forward to your talk.
Thank you very much, Janina, for the kind introduction. And also a warm welcome from my side to all participants of the seminar. As you have already heard in the lecture of Mary Grace, adaptive optics is a powerful tool to get the best performance out of a STED microscope. This is, of course, not only true when working with super resolution, but if we are working at the highest achievable resolution, we want to avoid all potential perturbations to our experiment. In my presentation, I will deal with the question what adaptive optics is already capable of today, and to what extent it can be used in biomedical research in the future without the user necessarily having special optical knowledge. Aberrations are not a phenomenon limited to microscopy, but we can also encounter them in daily life. At the moment, it is certainly not the first choice for many of us to go to the beach or the swimming pool. Nevertheless, we can observe very well what influence aberrations have on visual perception. Finer structures can no longer be exactly recognized, and the picture is overall distorted. Microscopes are beautiful instruments. All optical elements are manufactured with a precision of a few 10 nanometer, and therefore it's not surprising that the sample itself, which is also part of the optical path, can influence the imaging performance. And the main effects we can nicely see at a graph I took from a chapter I wrote together with Stefan Hell about 15 years ago for the Handbook of Biological Confocal Microscopy. And we see if we image in a strongly aberrated sample as compared to a non-aberrating sample, the brightness, the overall brightness is reduced, and also we get a decrease in resolution, which we can see here from the measurements of the point spread function. So we have a decrease in resolution and we have a decrease in intensity. And this image was, or those data were recorded with a normal wide field microscope. So this is true for all kinds of microscopy, but when we are working with super resolution, the problem is increased because the resolution we can obtain also depends on the intensity, either with the intensity of the detected fluorescence, like in Palm Storm, or the intensity of the depletion beam, like in STED microscopy, which means that we not only get a decrease in resolution due to the optical performance of the lens, which we have here, but we get an additional decrease in resolution due to the decrease in intensity. Therefore, we also formulated in this chapter practical strategies which can help to reduce refractive index mismatch and therefore to reduce aberrations and get the best performance out of the microscope. And those strategies were to use appropriate objective lenses, to avoid situations in which the sample is thin but far away from the cover slip, and to observe fixed specimens only in refractive index match systems, which is of course very difficult to do when one deals with living cells or living samples. Therefore, we also recommended that if the experimental settings do not allow for aberration minimization, one should think about aberration balancing or aberration correction. At that time, aberration correction was not as straightforward as it is today, because the devices were not available. But today things have changed, and there are devices which are not too expensive and which can be nicely used for aberration correction and can be implemented into microscopes. So for example, in my lab we are using deformable mirror, which you see here, which is just placed before the scanner of this STED microscope, and the deformable mirror consists of 144 actuators, so 144 elements, and it has a stroke of 3.5 microns, which is more than 4 wavelengths, and it is pretty fast, so it has a response time of 100 microseconds, which is in the order of the time we need to record only a few pixels. So compared to the time we need for recording a whole image, those elements are extremely fast. When we are using adaptive optics in microscopy, an important question is, of course, how deep can we image into an aberrating sample? And in order to answer this question, we can perform a very simple experiment. So we use a microscope. So here's the objective lens, a proper immersion medium. And then we use a sample which consists of 2 cover slips. And both cover slips are holding fluorescent microspheres, which we use to measure the point spread function we can achieve at a certain imaging depth. And between the 2 cover samples, we just fill in media of different refractive indices. And here's one example, or 2 examples of 2 measurements. So what we used here is an oil immersion lens, which we see here from the refractive index of the immersion medium. Then the 2 cover slips were spaced by 11 microns in case 1 and 29 microns in the second case. and the medium we filled in between the cover slips was mainly PBS buffer, which has a refractive index which is close to water. And what we see that we have the optimum optical performance in both cases if we are just imaging beneath the first cover slip, but if we go deeper into the sample, we get aberrated point spread functions. So already when we image 11 microns into a watery sample with an oil lens, we already get a severely aberrated point spread function, but this aberration can be corrected for by using the deformable mirror, and we can recover the resolution we also get just below the first cover slip. And this is also the case if we image deeper into the sample, which has the same refractive index like this 29 microns here. We can also mainly correct for all the aberrations. And then we can not only perform 2 of those experiments, but we can perform many of those experiments. So we can use either an oil immersion lens or a water immersion lens, and then we can prepare many samples with different spacing, and we can always fill in mounting media which exhibit a different refractive index, and then we can always correct at the certain depth for the aberrations. And the nice thing what we see that for each medium we have a linear relation between the amount of aberrations we have to correct and the imaging depth, which is very nice, because this means that we can already predict how much aberrations we would have to correct for if we know the refractive index of our sample. And also because we have this linear relationship and we know the maximum stroke of the deformable mirror we are using, we can also say what the limit of our imaging system is. And this is shown here. So again, we have the water lens and we have the oil immersion lens, and the water immersion lens has a certain maximum focusing depth, so we can't focus deeper into the sample. And we see that over a large range we can correct with our mirror for the aberrations. If the refractive index mismatch gets too large, then we will not be able to correct for the entire aberrations if we are focusing too deep into the sample. So this would require a deformable mirror with a higher stroke, but still we can always image at least something like 120 to 130 microns within the sample. And at a certain refractive index mismatch, one would have to switch over from the water lens to the oil lens, and then at the oil lens, or again over a large range of refractive indices of the mounting medium, we can image up to the full depth, imaging depth of the lens. And again, if the delta between the refractive index of the immersion medium and the mounting medium gets too large, we still cannot image to the entire depth, but again to something above 100 microns. But this results really indicates by using adaptive optics, one is able to image all kinds of different samples by only using 2 different objective lenses, which is very, very nice, because then we just need a microscope which has 2 high-NA lenses, and we just have to know what the refractive index of our sample is, and then we choose the objective lens which is more suitable for the samples, and all the other distortions or the residual distortions we get from the samples can then be corrected by using the deformable mirror. Of course, one can correct for the aberrations in a manual fashion, like we did in the previous experiment, but of course it would be much better if one would have something like an autopilot for the aberration correction, which means that the aberrations will be corrected automatically without expert knowledge of the user using the microscope. And one way doing this is, of course, using a Hartmann check sensor, like we saw in the previous lecture. But Hartmann check sensor, when we are working with fluorescence, has some shortcomings because we have to distribute the fluorescent signal over a large area, because a Hartmann check sensor is working with the micro lenses, and it always has to image certain parts of the wavefront. So it means we need a lot of fluorescence to get a decent image to analyze the aberrations. And of course the aberrations or the fluorescent photons we use for measuring the aberrations cannot use for imaging anymore. And especially nowadays, when we are talking about live cell experiments and long time experiments, all of us know that the fluorescence photons are very, very valuable, and that we want to use as many of them as possible. Therefore, it would be ideal to be able to correct or to measure the aberrations while recording the images. And there's a certain state of the art in the field which I just want to explain now. So what can be done, that one uses a deformable mirror, then changes for every image which is recorded slightly the kind of aberration, and then measure a metric which depends on the brightness within the image and also on the sharpness within the image. But the problem is, because the metric also depends on the sharpness, which means that we have to image the same plane within our sample very often consecutively in order to determine the optimum aberration correction. And this again means that we lose a lot of fluorescent photons. And this means that the shortcomings of the state of the art are that we are slow due to the iteration over the images, because we have to record many images in a row in order to optimize the metric. We are prone to photo bleaching even before we start the actual measurements. And the metric depends on the sample brightness and on the sample structure. And in an ideal case, what one would have is something like a photon stream based metric, so that we don't depend on the structure of our sample when we determine the best aberration correction. And that's the vision which we had. And just to show you how this should work, so our idea is that we just change on the aberrations while we are imaging, and that the microscope, like a real autopilot, is already starting correct for the aberration while the image is recorded. In order to explain the basic idea of the metric we want to use, we just have to switch back to one of the first slides which I showed, and we know that aberrations are decreasing the overall intensity, and this means that aberrations also decrease the local STED intensity. And this means that if the local STED intensity is changed and we use the STED light for depleting the fluorophores, that aberrations also affects the photophysics of the dye. And this means if we look at the arrival time of the individual photons while imaging either under aberrated or not aberrated conditions, we see that, for example, here we imaged a sample where we stained for vimentine, that just beneath the cover slip we see a certain distribution of the arrival time of the fluorescence photon just after each STED pulse. If we image deeper within the sample, then we get aberrations, the local STED intensity gets reduced, which means that we see more fluorescence photons at late time points after the STED pulse. And if we correct for the spherical aberrations, we see again that we get a reduction of the late photons, and now then again we get more early photons than late photons. This means that we actually can use a very simple metric. So if we just look at those histograms of photon arrival time, that we just use as a metric the number of photons we detected during a certain period after the STED pulse, so which we call the early photons, so just photons which arrived up to a time point t1 after the STED pulse, and this amount of photons is just divided by the amount of so-called late photons, so all the photons which arrived after T1 up to a certain time T2. And this metric doesn't know anything about the overall brightness of our sample, because we are just using a ratio of photon numbers, so it doesn't depend on the overall brightness, and it also doesn't know about sharpness or the structure of our sample. We just have a photon stream, and we just look how is the arrival time within this photon stream with respect to the STED pulse. And now we just can use a sample and just can calculate the metric for certain aberrations. So here, for example, we performed experiments where we just introduced astigmatism with our deformable mirror to a sample containing fluorescent microspheres just below the first cover slip, and we nicely see that the metric has a maximum for the situations of zero astigmatism. So that's what we expect, because we are imaging just below the cover slip, so just at the surface. And we also see that when the metric has a maximum, we see the maximum resolution in our experiment, and we also see the maximum signal in our experiment. So this means that the metric really tells us whether we are imaging under the optimum conditions. And this is not only true for the astigmatism, but also for other kinds of aberrations. So here are just examples now of different metrics, again just for the same sample. So It's the same if we correct for coma, or it's the same if we correct for spherical aberrations, or it's also the same if we correct for trefoil. And here we also see, okay, that we even have to correct a bit for trefoil just below the cover slip, which means that our microscope was not working under optimum conditions, but close to optimum, but we could still improve our microscope by correcting a bit for the trefoil aberration. As I said before, what's very nice about this metric is that it is very stable against bleaching, and it doesn't depend on the sample brightness. So to show that, what we did, we just took the same sample and imaged it many times in the STED mode. So here we see 4 out of a series of 30 images. We see that the sample is bleaching over time, but if we calculate the metric, we always get the maximum at the same amount of compensated spherical aberration, which means that we just have to maximize the metric. And by doing that, we always get the best optical performance, and we don't have to take care or consider bleaching or other effects due to the sample. And in order to demonstrate that the metric is really not depending onto a structure within the sample, we use 2 different kinds of samples. So the first sample was just a sample consisting out of 40 nanometer sized nanospheres, and we introduced spherical aberrations by our mirror, and we just saw that if you're just imaging below the cover slip, that we get optimum performance if we do not introduce any additional spherical aberrations. And if we follow the metric, we get the same behavior if we are using a sample which is not containing fluorescent microspheres, but it's just a solution of R2647N in PBS. So there's no structure within the sample, but still we see a difference in the photon arrival time, which means that we really just have to look at the photon stream, and we don't need any structure, any property of the sample, because actually a sample just consisting out of a solution of a fluorescent dye is actually the worst thing. So there's no way to figure out whether the aberrations would have been corrected correctly or not, because there's no structure. But again, under such unbeneficial conditions, the metric also performs very well. So what about a practical example? So how does this metric really perform under real condition, and how close is this strategy for correcting for aberrations with respect to the vision I told you a few minutes ago? So in order to figure out that, we just labeled the microtubular network in fixed cells, and then we just looked at our photon stream while we are scanning. And you see, so this is recording in real time, what happens that while we are scanning, we are analyzing the metric, and the microscope slowly adapts to the optimum imaging conditions. And at a certain point, so this is where here I have the green bar, we have optimum imaging condition, and we get the best performance out of our microscope. And actually here we were not working at the optimum signal to noise ratio. Of course, we are analyzing those photon arrival times, which means that we always need a certain amount of photons in order to take a decision. And if we reduce the amount of photons which we count before we take a decision, you can see that actually the correction can be performed much faster, which is shown here in the sample. So this is exactly same sample, same field of view, and you see that the whole system just needs a few scanned lines to find out the optimum scanning, the optimum aberration correction conditions, and the rest of the frame is then imaged under perfect conditions, although the aberration correction autopilot is running the whole time. And just to give you another example, again, metric running with a high signal to noise ratio, you see the system is adapting to the optimum imaging condition, and at a certain point the system is locked, and we are just working with close to perfect conditions. And again, if we just use the minimum amount of photons to take a certain decision, again the system locks in much faster. Again, it's just maybe 1/10 of the frame which we need to correct for the aberration, so we don't have to image over and over the same area in the sample again and again. We just start scanning, the system locks in, and we work with optimum aberration correction conditions. This brings me to the end of my talk, so I hope that I could convince you that adaptive optics is a powerful tool for getting the best performance out of a microscope, not only a super resolution microscope, but especially a super resolution microscope, and that in STED microscopy we have the unique possibility to build an autopilot for aberration correction, which means that the user can really concentrate on his or her experiment, and that in the future samples will not have to be adapted to the microscope, but that the microscope will adapt to the sample. But of course, I don't want to end without acknowledging the people in my lab who actually performed all the work I presented you today, especially Claudia Geisler, Francesco Rocca, and Depatrita Ghosh. And I also want to thank the funding agencies, especially the German Research Foundation, the State of Lower Saxony, and the Federal Ministry for Economic Affairs and Energy. I also want to thank you for your attention, and I will gladly now answer your questions. Thank you.
Thanks a lot, Alex. Great talk. We got a lot of questions, so let's just start right away. How does one determine T1 in the metric?
Yeah, so of course T1 is a function of the length of the STED pulse and also of the lifetime of the fluorophore. But actually, a good guess is always to make T1 identical to the length of the STED pulse, and with that you are totally fine. And yeah, so that's mainly it. And if you adapt it a bit more finer, then maybe you gain another 5 to 10% of precision.
All right, thanks. Then the next question is about lifetime-based adaptive optics. I understand that it's faster than image metrics based because you don't need to wait for the whole image before correcting. How does the number of test corrections that you need compare to image metrics based? Is it the same?
Yeah, so actually it's a metric, and like also the other methods, we always have to wiggle through the aberrations. So the number of aberrations we have to test are the same. But maybe one thing I have to mention is, of course, we are just wiggling a bit. So actually, the aberration correction is also on for the rest of the frame, and it would also adapt if things change over time. So actually, it's a bit faster when we are close to optimum conditions. But anyhow, we have to go at least 3 times through every order of aberration.
All right, thanks. Next question. Do I always need a small piece of every cell to find out the metric? So can I ever image a whole cell without a few scan lines where it is still adjusting?
So as I said before, so it depends, right? So if you're already very close to optimum conditions, you just can image the cell, so it will not move out of the optimum imaging condition. But if you are far away, then you always need the lines, because at some point you always need to get the information. Of course, it's also fine if you just pre-scan a bit and then jump back, and then you can record the whole line. So that would be also possible.
Mm-hmm. Good point. All right, final question for you, Alex. How is the day-to-day stability of the deformable mirror used?
Yeah, so actually, so the one we use, it's very fine. So we just installed it one time. Then, of course, we had to calibrate it, and then we haven't recalibrated the whole system for the last 4 years. So from my experience, so it's really, really solid. So you just have to install it, and that's it.
Cool. Sounds great. Perfect. So thanks a lot, Alex, for your time and for joining us for today's session. And next, I'm more than happy to introduce to you my colleagues Matthias Reuss and Jan-Gero Schloetel, who will first introduce us to the abberior instrument's adaptive optics solution and then show you live on the microscope how to acquire a STED image with adaptive optics optimization. Matthias is our head of research and development at Abberior Instruments, and he is a physicist by training. During his PhD work in Stefan Hell's lab in Heidelberg, he developed the EasySTED face mask, which is a central part of our Steadicon microscope. Afterwards, he joined the Skylife lab in Stockholm for a postdoc, and already during this period, he co-founded our company, Abberior Instruments. Since then, he worked on several different aspects on the company, from sales and marketing, for example, to research and development now. And Gero is one of our application scientists at Abberior Instruments. He has a profound background in biochemistry and biophysics, and during his PhD in Glasgow, he developed a fluorescence-based DNA assay and then joined University of Bonn to supervise a STED microscope facility. Since almost 4 years, he works now in our team, and we are extremely happy to have him on board. Matthias and Gero, I'm really looking forward to your presentations. The stage is yours.
Thank you, Janina, for the kind introduction. Many thanks also to Alex and Mary Grace. Before I hand over to Gero, I'd like to say a few general words about adaptive optics and how we implemented it in our microscopes. What you see here is a schematic view of a laser scanning microscope, a STED microscope. So we have STED lasers being directed towards the objective lens and the sample. We have excitation lasers for 1 or 2 photon excitation, and of course fluorescence comes back from the sample and is directed towards filters, pinhole, and detectors. Now, in order to enable adaptive optics for this system, there are 2 key components that are required. First, we have a spatial light modulator, which sits in the beam path of the STED lasers. And then we have a deformable mirror, which is located in the joint beam path. The role of the spatial light modulator in our microscopes is twofold. First, it provides beam shaping for the STED beams. That means it applies a certain phase distribution to the STED beams, so that the STED beams focus to a hollow donut or bottle beam in the sample. And here the dynamic nature of the spatial light modulator enables us to adapt those phase distributions to the different objective lenses. In particular, different diameters of the back aperture of different objective lenses. And that enables us to do 3D STED with virtually any objective lens that is out there and has reasonable quality. And the second role of the spatial light modulator is to provide aberration correction for the STED beams. Then we have the deformable mirror. All beams, STED lasers, 1-photon excitation, 2-photon excitation, fluorescence, all beams pass the deformable mirror. That means that aberrations can be corrected for all beams using the deformable mirror. And this is really what enables imaging of difficult samples. Gero is going to show you tons of examples for this. I like to sum up the key advantages of our adaptive optics solution for you. First, the correction of spherical aberrations follows along with the focus depth. So as focus changes, either because the user moves the nose piece or the system moves focus during a volume scan, the amount of correction, the spherical aberration, changes in order to make sure that the imaging conditions for each depth, each focus depth, are optimal. On top, we can correct arbitrary aberrations, because arbitrary aberrations can be displayed on the SLM and the deformable mirror. It can be astigmatism, trefoil, you name it, or any combination of every aberration. And this, again, this is what enables imaging of difficult samples, samples that are inhomogeneous, samples that are index mismatched. Typically, the limitation really is the working range of the objective lens. Our adaptive optics system is upgradable. So if you already have a microscope from Aberria Instruments, but no adaptive optics yet, you can easily get one. It's a field upgrade, very simple. Very important, neither the SLM nor the deformable mirror has moving parts. And this is in contrast to, for example, correction collars. Correction collars are macroscopic mechanic appliances. When they move fast, they can cause vibrations. They can wear out. When lenses are moved, it can cause misalignment of optics. None of that is a problem with SLMs or deformable mirrors that we use. The non-mechanical nature also means that they're fast. The shape of the deformable mirror can easily be changed for every scan line. And this is in fact what enables the correction of spherical aberrations during a scan at different focus depths. And finally, I mentioned this, with the SLM, we can do STED with any objective lens that is out there and has a reasonable quality, be it an oil objective, water, air immersion, dipping lenses. We use objectives with long and short working distance, silicon, glycerol, you name it. This is it for me. Thanks for attending, and I hope you continue to have fun.
Thank you, Matthias. My name is Jan-Gero Schloetel. I'm an application specialist at Aberria Instruments, and I'm going to talk about the biological applications of adaptive optics. Since we are going to talk about 3D STED a lot today, let me give you a quick introduction. In any confocal image, the resolution is limited by the size of the focal spot, or PSF, point split function. In a normal confocal system, that PSF will be roughly 600 nanometer in the z-axis and about 200 nanometer in x and y. So any structure smaller than that won't be resolved with a large scanning spot like this. In 2D STED, we can shrink the size of this spot to 25 nanometers in the XY axis. However, in the Z axis, it remains just as long as the confocal PSF. In 3D STED, we can shrink the PSF also in the Z axis, but on the other hand, it gets slightly larger in the XY axis again, leaving us with a circular isotropic PSF, which will resolve structures better in X, Y, and Z. This is how this would look like in a biological structure, here a nucleus, and we have stained nuclear pores in green and lamin in red. And you can see in this image, which is a vertical slice or XZ view, you can see in 2D STED we can't really resolve these two levels which are on top of each other, but when we go to 3D STED we see the large empty space in between because we now have an improved Z resolution in this image. 3D STED can also help if you look at very, very small organelles like mitochondria, and if you want to look specifically at something which is on the outside membrane, then better optical sectioning, as with 3D STED, will help you to see these rather than seeing everything which is also on top and on the bottom, like in the confocal image. A really important application of 3D STED is obviously volume imaging, because now you can see these structures with a better resolution in all dimensions, and you can tell if structures are simply on top of each other or if they're connected, and so on. Here we did a 3-color 3D STED image, and you can see the difference to the confocal image in the rendering. What is important about 3D STED is that it can be affected by refractive index mismatches, and when we're talking about that, we can see this very easily on the example of, say, using an oil objective, where the immersion oil has a very high refractive index, but if we then image, for example, into a water sample, we have here a much lower refractive index, which is a refractive index mismatch. Doing so, we could image, for example, a bead layer here very close to the cover slip, very bright still, even though there's a mismatch. And the 3D STED image is also very bright when we are really just at the cover slip level. Going deeper into the sample though, the mismatch will cause aberrations, which make the image much, much dimmer in confocal mode, and we have seen this in normal confocal microscopes as well. In 3D STED mode, we can see that there's nearly no image left at this depth. And as normal, 3D STED is much more easily affected by these mismatches than the normal confocal image. What can we do against that? Well, for starters, we can choose the right objective, say a water objective to image into water. In this case, we can see at the higher depth, here 13 micrometer roughly, the confocal image is still bright, and also the 3D STED image has still the full brightness and is sharp. Now, oil and water are not the only media we can find, and there's a lot of objectives to choose as well, which you can use to match to these various or mounting media. Very popular is the oil objective still, and many of the commonly used mounting media like Marymount Solid or Prolong Gold, for example, will not really reach the refractive index of the oil, which means there's a mismatch, and going deeper into the sample, you will have a loss of intensity and a loss of 3D STED. In water, there will be cultured cells or less dense tissue that works really well. If you get denser tissue, you might want to switch to a silicon objective, but there you might still have to compensate for small mismatches between the immersion silicon oil and the tissue. Also, you need to know that for many of the curing or mounting media or media which need to dry to harden, you will find that depending on the time you use for curing, you will have different refractive indices in this medium, and again maybe a mismatch to the immersion liquid. When we're talking about samples, I just would like to mention quickly that nowadays there's lots of dyes you can use for STED microscopy, and if you're interested in this, you can have a look at our webpage or contact us. Now, so far we have been looking at images of single cells. This is not very deep, and when you go to tissue, we go much deeper into the sample. But that brings challenges with it as well. For starters, the deeper you go, you get more absorption of the lasers and the fluorescence, but also scattering. What we do is we use pulse high power lasers and very sensitive APDs for detection, so we get a bright image even if we have losses going deeper into the sample. But we don't stop there. What we have developed is adaptive optics, and we have two types there: the spatial light modulator, which is what we use in our Easy 3D module to do 3D STED. An add-on would be the deformable mirror, which I will explain following. If you look here at a focus, ideally in the focal plane, all the lasers should be focused into one spot, and the fluorescence light would do the reverse way here. However, if you have an aberrated focus, because we're going through tissue, then we don't have an ideal spot here, focus spot, and also on the way back we lose fluorescence light. But we can go from this aberrated focus to a finely focused spot by using our deformable mirror. In detail, basically step one would be using adaptive optics, which are already included in our Easy 3D STED. And as I mentioned before, first we check the sample for a refractive index, we pick the right objective lens, and here Easy 3D STED is very much at an advantage because we can choose the objective lenses. This is not true for all STED microscopes. This is something we can do especially because we can adapt to the aperture which each objective has, and objectives have different apertures at the back. So our STED patterns on the laser are adaptable, and we can use a large range of objectives, so we can adapt to a large range of mounting media and get a good image. What the spatial light modulator in Easy 3D STED can then do is the residual mismatches between the mounting medium and the immersion liquid can be compensated. And that works for the STED laser, so we get a very good 3D STED image, for example. It doesn't affect the confocal image though. To do so, we have also developed a second method, which is the deformable mirror, which can now act on the excitation and detection as well. So it's not only the STED laser, not only the STED mode is improved, but also the confocal image in both ways, detection and excitation. So this gives us good 3D super resolution, bright images even at a high sample depth, and also the samples are, or the images of the samples are less distorted. An example where we go into tissue would be here in a sample of the inner ear of the mouse, and we are sampling here the hair cells, the inner hair cells, and this is a Z slice or vertical slice again, and we can see the improved 3D resolution in this image. I would like to thank Christian Vogel from UMG Göttingen here for the sample. Now, if we go to a tissue sample which is really large and we want to image all the way to the bottom of the sample, we can look at, for example, this cleared sample of kidneys. Cleared because otherwise we would probably lose a lot due to scattering. However, we can go really nicely, very deep into this sample. Kidney tissue otherwise would scatter a lot. We're using here again an oil immersion objective. The mounting medium has a slightly lower refractive index comparable to some of the hard setting media. What we can see here is when we go into the sample in a conventional 3D STED mode, we can see in these squares here, the image deteriorates really quickly, and only at the very top we get a good image. Using adaptive optics with Easy 3D, we can see the image quality is sustained even down to 60-70 micrometers into the sample here. I would like to thank David Unas UES for this sample. Here's a 3D rendering of the sample where we collected whole volume using Easy 3D and also our adaptive illumination technology. Now, what we also can notice is though that the confocal image going deeper also gets dimmer. And if you look at the very top here, we can see that there's actually quite a bit of intensity loss going from the very top to, say, 20 micron and then even deeper. And that's the same even if we have Easy 3D on, because that really only compensates for the STED mode. And there we have a better solution. We can use the deformable mirror, which is the step 2 of our adaptive optics modules. And we can see that when we are imaging here in a fly sample at about 100 micrometer depth, you can see the confocal image in an uncorrected image is dim and also distorted, like stretched in the z-axis. However, if you look with correction or with a deformable mirror, we can actually recover the brightness and the distortion is also not noticeable anymore. And this effect is even stronger for Easy 3D STED, where we see barely nothing in the uncorrected image, and with Easy 3D STED and the deformable mirror, we can see a lot of fluorescence, and we also have an increased resolution, seeing, for example, this circle here, where it's just some smear in the confocal image. Again, we're looking at an XZ or vertical slice here. I would like to thank Mr. Schnorrenberg from the AMBO in Heidelberg for this sample. Now, at this point, I would like to start with a practical demonstration of the deformable mirror and how we use this to get this sustained brightness of a sample at great depth. Here you can see the expert line software interface with the Easy Commander. Easy Commander lets you pick dyes and it will automatically set parameters for these dyes. You can also manually set those parameters and define channels using a live dialog. And there you can set things like excitation lines, exact detection windows, dead lasers, time gating. An even easier way would be to open a template, and that lets you start a measurement exactly the same way as you saved it before, which allows you to repeat measurements with the same settings. This template has several windows. Some of them are overview windows, some are measurement windows, like there on the bottom. Looking at an overview window, we can see kidney tissue. This tissue has been stained for actin using aberrations star red phalloidin, and it's been embedded in aberraria mount liquid. And we're using here the water objective. Going from the XY overview, we start a XZ overview, basically a vertical overview. And here you will be able to see the thickness of this kidney tissue. It's a tissue slice. It shrunk a bit since the last demo, but it's about 30 to 40 microns deep. In this sample, the aberrations increase with sample depth, and the deformable mirror will compensate according to depth. Now we start a scan, and this will indicate where the cover slip is. The bottom here is a reflective image, and at the cover slip surface, the reflection is the strongest. Once I've found the surface, I go to the deformable mirror menu, and there I can click Set Cover Slip Level. Once the cover slip level has been defined, the software always knows how deep we are in the sample. I'll rerun the vertical or XZ overview, and I want to select a region of interest, like so. And I want to rerun this region with finer pixel. For that purpose, I drag and drop that region into a new window. When I start this new window, it will start a scan with finer pixelation. And I can increase the size of this window and keep running this scan in normal mode. This is again a vertical view You can see the lower regions appear less sharp, and this is what we want to improve with the deformable mirror. For this, I go to the deformable mirror menu, and I simply press Auto Follow Depth. Now you can see the lower regions are much sharper, brighter even, and that's just the effect of the deformable mirror. While scanning, I can switch it on and off again. Now it's much dimmer and more blurred. And now it's much brighter again at the deeper levels of the sample. Now at this point, I would like to switch to 3D STED or 3D super resolution, because this is where the deformable mirror has probably one of the biggest effects. I will simply select a region around here and drag and drop this one into the next window. You can see for the next window in Easy Commander, STED is activated and the 2D 3D slider is all the way to 3D. Before I start the scan, I'll go to the deformable mirror menu and I will deactivate the automatic compensation. When I start the scan, you will see on the right the STED image or 3D STED image, and on the top you can see there's a very sharp structure, and compared to the left side where the confocal image is, the resolution on the right and the 3D STED image is much higher. In the middle, the image quality suffers, and very deep on the bottom there's much noise and very little signal. I will duplicate and rerun this image with exactly the same settings, but in the deformable mirror menu I will activate auto compensation of aberrations. Now rerunning the scan, you can see on the right side the 3D STED image, but this time aberrations have been compensated by the deformable mirror, and you can see the brightness and the 3D STED resolution are maintained all the way down really deep into the sample. When you compare the 3D STED image on the right side to the confocal image on the left side, you can definitely see more structural detail in the 3D STED image. And that's all thanks to the deformable mirror. In this image, the deformable mirror had to be actually very fast because we're doing a vertical scan here, so it's an XZ scan. And basically every line in the image is a different depth level. And the deformable mirror has to adapt its shape in every line to do a different level of compensation of aberrations. So I think we get a pretty good result. And in this sample we were looking at kidney tissue mounted in abberior mount liquid, and we were using a water objective, and we could maintain good resolution all the way through this tissue slice. But we've done lots of different samples before, and I will show you a few different combinations going back to the presentation. There we go. Now, we were looking at kidney tissue, and it was embedded in abberior mount liquid. If we compare this with this chart here, we can see abberior mount liquid has a refractive index below 1.4, while the water objective optimally works at a refractive index of 1.3. So there's a slight mismatch between the refractive indices, and that explains in part the aberrations we were seeing, which was reducing the image quality at the deeper sample levels. Another factor here is that we were imaging in tissue, which is also increasing the refractive index above water. So both parts here contribute, but we were able to compensate that very well with the deformable mirror and got a good image quality at the bottom of the tissue slice. Now, looking at the chart, you would expect the silicon objective to perform slightly better with these samples because it has a refractive index in the immersion liquid, which is closer to the sample medium. And I've tried this before with the same sample using a 100x silicon objective, and you can see on the top here the image quality is good, and going down deeper into the sample, the image quality doesn't decay as much as with a water objective, meaning the PSF here is not stretched out completely at the deeper sample levels. However, switching on the compensation with the deformable mirror, you can see that the image quality is improved at the deeper levels of the sample. What's interesting is if you go back to the uncorrected image, is at the top you have good quality, and switching on the DM, not much changes at the top. And that's important because the deformable mirror adapts to the depth of the sample, so at the top it will do no correction at all, and going deeper into the sample it will correct very strongly for the aberrations. Go back to uncorrected, you can see there's a big difference, and that's important, and for that the deformable mirror has to be very fast acting to compensate at age level the right strength. Now, going to oil objectives, it is a bit harder to find a mounting medium which will guarantee that the refractive index of the mounting medium is matched to the immersion liquid. But there's, for example, prolonged glass, which is very closely matched to the refractive index of glass and the immersion oil. You can see imaging into this sample here in prolonged glass, which is again kidney tissue, same stain, but in prolonged glass. You can see the image quality is quite good, still at the bottom. However, switching on the DM, you still gain quality. This may be due to me probably not curing this mounting medium as long as I should have. However, it always helps to have the deformable mirror here to compensate any residual refractive index mismatches, especially if you're using a oil objective which doesn't have a correction color. However, correction color, as opposed to the deformable mirror, can only compensate at one level, while the deformable mirror can actively adapt to the level or the depth of the sample. Now, what I've also tried is using kidney tissue which has been cleared, and you can image very, very deep into these samples. And what we can see here is an image recorded with a 100x oil objective, which has a working distance of 100 micrometers, and you can see we were able to image all the way to the working distance with this sample. What's interesting is that we're here looking at a 3D STED image, which you would expect to decay in a sample if there's a refractive index mismatch. So as I mentioned earlier, 3D STED is very susceptible to refractive index mismatches. But we used the deformable mirror here, and we could maintain image quality very much going deeper into the sample here. And you can see very fine details here in these structures. We're looking here at podocin and nephrine stained by David Unes US. Thank you to David for this nice sample. On the right side, we can see the uncorrected image, which means we have not used the deformable mirror to compensate for aberrations. And really at 20 micron in the sample, there is the limit for good quality 3D STED imaging. We've acquired these images actually at the transient microscopy workshop this year, and we had an audience who could watch this deformable mirror in action there, and I hope we can do this again at a later workshop as well. Here I'm showing you an image of a Drosophila gut stained for tubulin, and this time we are looking not at a vertical XZ slice but a normal XY slice, as you probably mostly used to. And here's the uncorrected image, and we were here imaging at about 40 micron depth. Switching on the deformable mirror, you can see the image quality increases and we have more brightness and the image is sharper. Since the structures are fine, we zoom in a little bit and repeat this, and you can see the uncorrected image here, and switching on the deformable mirror, you can see an increase in brightness. And I think by now I've covered most parts of the deformable mirror, looking at XZ slices, where you can see the biggest differences, and 3D STED, which gets enabled even deep in samples using the deformable mirror. And here we're looking at an XY slice in confocal mode, which can also benefit from the deformable mirror. And with that, I would like to finish my presentation, and we can start the question and answer session in a minute. Thank you very much.
Thanks a lot, Gero and Matthias, for this great presentations. I'm always quite impressed on how much better it just looks with the adaptive optics system turned on. And we got a lot of questions for your part. First question is, what is the maximum depth with the DM?
The maximum depth with the deformable mirror is essentially limited by the working distance of the objective lens. It's similar to what Alex showed in his presentation. If you have a water immersion lens and you focus into water based embedding medium, you essentially can go up to the working distance of the objective lens. The same holds for an oil objective lens, and there's a zone in between where you would need, or it would be good to have a glycerol objective to go all the way to the working distance of that as well. But through the whole range of refractive indices from water to oil, you basically have always at least 130 microns, roughly.
Cool. That's a longer question here. Can you please delineate what's included in the standard EZ3D STED? My understanding is there is adaptive illumination and adaptive detection, which might be the optional item. How much of this can be done with just adaptive illumination if that's the standard package?
The EZ3D STED essentially is the spatial light modulator. So that besides beam shaping for STED, that gives you the possibility to correct for aberrations in the STED beam. And with STED, that already goes a long way towards optimal imaging. If you do confocal imaging, obviously having an SLM, an EZ3D STED in the STED beam path does not help. So that would be a case for deformable mirror. And of course, also the deformable mirror adds a lot to STED imaging. Adaptive illumination is a whole different thing. Adaptive illumination is a way to adjust the strength of the illumination, so the intensity of the excitation and the STED beams to the underlying structure, so that you only put light into the sample where it really has an effect and where it's needed. So adaptive illumination is about manipulating the intensity of the beams based on where you are in the scan and where your structure is or not. And adaptive optics is about, well, what you just heard, shaping the wavefronts of the beams in order to correct for aberrations.
I guess during the last webinar series, the live cell STED webinar, we will show adaptive illumination on our microscopes. So please register for this if you would like to learn more about this. All right, next question. Does the deformable mirror improve imaging in brain tissue?
Yeah, I would like to answer this. So in brain tissue, it definitely helps. First, if you work in aqueous medium there, the brain tissue will have a different refractive index because of the higher lipid content. So the DM can help there and correct these or compensate these aberrations. What's also commonly done with brain tissues, I think, that you mount it in a high refractive index medium, and then you would work more with the oil objective. And this is done because the transparency increases when you use a high refractive index mounting medium. And with the oil objective, you then have the chance to adapt exactly to the refractive index of these high refractive index mountings. And that's really improving then the depth you can reach and the 3D stat quality.
Thanks, Gero. Next question. What percentage of light or signal is lost by adding adaptive optics to a system? That is, if imaging at the cover slip most of the time, is it better not to have the extra element?
The amount of light lost on the mirror is a few percent over the practical range of wavelengths that you're using. So the visible range and also the near infrared and the range covered by 2-photon lasers. Next to the mirror, there are a few optical elements in the beam path additionally, but it never exceeds more than a few percent. And so very strict answer would be if you really stay at the cover slip, perfectly at the cover slip, you lose a few percent of fluorescence. But as soon as you move away from the cover slip in a not perfectly index matched sample, essentially you gain more by the deformable mirror than what you lose.
Thank you. Next question. Do I only need one objective and compensate everything else with the deformable mirror?
So I would like to answer this because I've been working with a lot of different samples and using a lot of different objectives. So I'm used to always picking exactly the right objective for the right mounting medium. But with the deformable mirror, we can really go away from the optimum refractive index. So that's nice. And I think Alex has described nicely how when you go and have an oil and a water objective, you can cover most the biggest area of refractive indexes which you will find in samples. He's also shown that in the very center there was a little dip where you couldn't reach the maximum working distance of the objective if you're very far away with the refractive index from the oil and the water objective. And if for these cases where you want to go really deep and you want to hit exactly that refractive index in between, then I would recommend getting a silicon objective, which is right in the center. And with this, you have really covered every imaginable mounting medium. And you can then go in all of these media to the very bottom. And with the DM, you can not only stay at the top or the bottom, you can really quickly move from the top to the bottom and cover all the or compensate at every level adaptively, automatically with the deformable mirror. So yeah, it helps. You probably need 2 objectives. If you want the top performance at the right at the center, maybe get a silicon objective, but you can cover most things already with 2 objectives then.
Great, thanks. Next question. Would the same performance be achievable also with the correction collar?
With a motorized correction collar, I guess if you do slow scanning, so you have very long lines, in principle, yes. I think for what Gero has shown, it wouldn't be fast enough. So there's a lot of mass you have to move around, causes vibrations if you move it fast, has inertia. So clearly the deformable mirror really, which is super fast on the order of 100 microseconds, is the much better choice. And for a fast scan, if you do, especially if you do XC scans, I doubt that you'll be happy with the correction collar.
Okay, thank you. And now the next and final question. Do you need to mount the tissue section directly on the cover slip to work for STED?
I could answer this. So no, you don't have to, because especially if you use a mounting medium which will permeate through the sample, you will have still a roughly linear compensation through the sample. And actually the top section of your sample will, if you mount it not right on the cover slip, already appear distorted or dimmer if you do this without the deformable mirror. And you will, from the start of at the top, then recover the original image as if it was mounted at the cover slip. So in essence, basically, with a deformable mirror, you don't have to mount anymore at the cover slip because you're not losing fluorescence when you're going deeper. When you get it wrong and it's sitting a little bit deeper, you have more tolerance there. You can actually don't have to attach it right away at the cover slip level.
Cool. So thanks, Gero. Thanks, Matthias, for joining us today for this webinar. And thanks also for all the questions answered. It's really great. So now we got so many more questions from you. Thanks a lot for this and for your awesome participation. Unfortunately, we couldn't answer all of them now, but we'll get back to you via email and answer your question. If you have more questions, please don't hesitate to contact us directly. And please also get in touch with us if you would like to demo our STED microscopes with adaptive optics with your own samples, either live with us in Göttingen or also remotely. This webinar was part of the aberrational Instruments Summer Symposia, so please don't forget to join us for the next events. Next week, our guest is Mike Heilemann, who will give a talk about STED paint, and afterwards, we'll show you on our microscopes how STED paint can help you to acquire high contrast STED images without any bleaching. With this, I thank you for joining us today. I hope you enjoyed our webinar, and see you next time. Have a nice day, stay healthy, and bye-bye.
Bye-bye.
Bye.